In this video we will discuss about this ITGA3 and how this ITGB1 helps in the immunosuppression. So far in the previous video we have understood that when we check the data from TGCA, that is the Cancer Genome Atlas, we are able to find out that these high expression levels of ITGA3 and ITGB1 is related to the low survival rate of the patient. So they wanted to check the hypothesis that whether this ITGA3 high levels of ITGA3 and ITGB1 will cause immunosuppressions and thereby helps in the pancreatic tumors progressions or not. So in order to check this hypothesis, what they did is they took the different pancreatic cancer cell lines, and the results that they are shown in this main figure is about PANC-1 cells and this MIA PaCa-2 cells. And for this ITGA3, the results are shown in the main figure. For ITGB1, we can refer to the supplemental figures. So first, they did use the CRISPR-Cas9 system to knock down ITGA3, and the results are shown here for the PANC-1. That we can see that the knockdown ITGA3 cadheranase has no signals, whereas the control has the signals. And these things they have confirmed using the mean fluorescence intensity, and also percentage of ITGA3 cells in this PANC-1 knockdown. So after validating that the ITGA3 has been knocked down successfully, the experimental plan is shown in the panel B, where what they did is they took the ITGA3 high cells and ITGA3 low cells and stained them with or labeled them with pHrodo dye, and then co-culture with monocyte derived macrophages and do the live cell imaging. So in this case, what they are able to find out, the high percentage change in the cytotoxicity is in the case where ITGA3 has been knocked down, whereas where ITGA3 is not knocked down, the percentage change is low. So this indicates that the ITGA3 helps in the immunosuppressions, and thereby the change in the pHrodo dye signals is less, whereas in the case of ITGA3 has been knocked down, in that case they are able to see the percentage change in the signals is quite high of this pHrodo, indicating that most of these cells are going to die. And the immunofluorescence image are shown in this left hand side of the panel C, where we can see that ITGA3 cadheranase are ITGA3 knockdown cells can be seen in the red in color. And once validating that their experiment, they're able to see a similar results that they have seen in this from the public level, that they have concluded or observed in the publicly available data, and they move one step further. So in this case, what it is, they took the control cells, and most of them are ITGA3 high, and then they use the column which is coated with ITGA3 antibodies. So with that column, they are able to separate two populations: the ITGA3 low cells, which directly pass through, and ITGA3 positive cells, which stick to the column. And these things they have validated again using the flow cytometer, where we can see that control cells are high, whereas ITGA3 low cells are low, our signals are slightly low. And these things, the mean fluorescence intensities are shown on the right hand side of the panel E, where it is clear that ITGA3 low cells signals intensity is low. And now they use a similar approach, that is they took the control PDAC cells which has high ITGA3 and the ITGA3 low PDAC cells, and label them with pHrodo dye and add macrophages and do the live cell imaging. And in that case also they are able to see that the percentage change in the cytotoxicities is high for the case where ITGA3 is low. And these things they have seen for MIA PaCa-2 cells and also for these PANC-1 cells. So that we can see that intensities are percentage change in the cytotoxicities are high for both PANC-1 cells and the MIA PaCa-2 cells. And although these results are not true for all the donors, but but the overall statistically difference is still exists between control and ITGA3 and control and ITGA3 low in both MIA PaCa-2 and PANC-1 cells. And the results for the ITGB1s is also found to be in the follow the same trend, and those results are in the supplemental figures. So this in all together indicates that this ITGA3 and ITGB1 helps in this immunosuppression, thereby promotes pancreatic cancer cells development. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.